So this is question 4, the plan, and it's a biology topic, effect of substrate concentration on enzyme activity. And so with any enzyme experiment involving substrate concentration, then the graph is sort of going to get, is going to look like if you increase substrate concentration, then rate of activity is going to increase. Then eventually it's going to level off as the enzyme active sites become full. And so your hypothesis is that as you increase substrate concentration, the rate of activity will go up and then level off. So as increase substrate concentration, then enzyme activity will increase, then level off. Then you sort of need a practical to do. Well, the practical that you did in class was that you took a potato, made a cylinder and put it in a pestle and mortar and ground it up with some water. That was your enzyme, and that was catalase enzyme. And then what you did was you got some paper discs. You dipped a paper disc in the catalase, and then you put the paper disc into the hydrogen peroxide it mentions. So that's your hydrogen peroxide. And then the disc sank and then rose to the top again when bubbles of oxygen formed, and you timed that. And that was because hydrogen peroxide, H2O2, used catalase, the enzyme, and that turned it into water, but also oxygen, which was a gas, and made it float. So the equipment that you are talking about, you're going to talk about potato, because that's got catalase, a borer to make a cylinder of potato, pestle and mortar to grind to a paste. You're going to need some water to dilute the enzyme, paper discs to soak with catalase. So can you see how we just mentioned in the equipment and then just given a quick one-liner, a few words to explain what you need. Then we need beaker for the hydrogen peroxide, stopwatch or stop clock to time disc to float. So that's the basics of the experiment, and you could spend as much time as you've got available explaining that. You have got our independent variable. So independent variable. What we're going to change, well, it says we want concentrations of hydrogen peroxide. So that is concentration of H2O2. And you could give a selection, so 1.0 molar, 0.8, 0.6, 0.4, 0.2 molar, and you could describe serial dilutions. So you could dilute this with water. So that's your independent variable. Dependent variable. This was your time for the paper disc soaked in catalase to sink and then rise again in the hydrogen peroxide. So time for disc with catalase to sink and rise in the hydrogen peroxide solution. Control variables. I'm a biologist, so I like controlled variables is a better word. So things that you'd keep the same. Then you would use the same potato for the same catalase for each experiment. So same potato for the same catalase. You use the same sizes of everything and volumes and everything, so same size or mass of potato, same volume of water, same volume of hydrogen peroxide, same size of paper disc, same temperature, same pH, because those will affect enzyme experiments. So plenty of control variables there. Risks. Hydrogen peroxide is an irritant. So wear lab coat and goggles, clean up spills, rinse hand in water if spilt. So basic risk assessment. And then the only last thing to do is think about data analysis. What would you do? So data, draw a graph. Well, you make a table first. Make a table. You could show the design of the table, what you do if you've got time. So this would be the independent variable, which is your concentration of catalase. This is dependent variable, which would be your time to sink and rise, and you've got your various values down there. The graph we've drawn up there, and that should be how you should treat data. So the basics of a plan there.